All right, let's go ahead and start again. Part 2, we just had another Understanding Ethylene Oxide just now. This one's going to be, this one's entitled Navigating Recent Standards Changes and Industry Challenges with Ethylene Oxide. So the main standard, ISO 11135, has just recently changed. The AAMI version of that standard's not quite out yet, so it's not published. So we'll talk about that. The ISO version is out there. It's not going to change from what the ISO version is. It's going to be verbatim. So the changes that are in there are exactly what we'll see in the AAMI version. We'll also talk about some of the gas blends got phased out, and that's created some issues with biological indicators. We'll also touch on, somewhat related to ethylene oxide, the duodenoscope issue with the superbug outbreak. There's a talk about possibly sterilizing those types of devices with ethylene oxide as a way to mitigate that problem. We'll just talk about there's a lot of other standards that are being revised as we speak, so there are a lot of them that are in transition. And then finally with ethylene oxide, there's issues with more and more devices that have batteries or stored energy, and ethylene oxide's flammable, so that can be an issue. So the ISO 11135 document, as we mentioned, it published in July of 2014, so last July, going on a year, just about a year ago, was when the EN ISO version published. So the US side is a little bit behind, and the reason why is that once AAMI decides to adopt the document, so they're going to take the ISO document, adopt it verbatim, but it also has to go through ANSI, which is the American National Standards Institute, get voted on. They do a public comment, and because there was some negative public comment votes, so even though the committee voted to adopt it, they have to address those public comments. They're not going to change the standard, but it took a little bit of time. It was supposed to have published by now. It was supposed to be out in January of this year, and now it's been delayed. And so one of the things with a lot of these standards, when they change, this is a big one because it talks about ethylene oxide sterilization validation. Normally there's kind of an unwritten rule that there's a 3-year phase-in period, that you don't necessarily have to have compliance for 3 years from the date of publication. That's what happened with the residual document, ISO 10993-7. There was kind of a 3-year phase-in period that was allowed for that. However, at the AAMI meetings this last May, just last month, the FDA came out and said that they were going to not allow a 3-year phase-in period, that they would require conformance with the document as soon as the AAMI version is published. And that's supposed to be published now. The schedule is September of this year, September 2015. And so some people may have to scramble a little bit. There's not a lot of huge changes on how you do the validation, but there are some subtle changes. We'll go through kind of some of the main changes that happen throughout the document. Again, this is the intent of this purpose was to merge the document into one single document. It used to be part 1 and part 2. Part 1 was the normative part of the section. That was where all the requirements were. Part 2 was the guidance to the document. This is where a compromise between the US and Europe. US, our position is we like lots of guidance, so somebody that maybe is not familiar with the standards can look at that, and the guidance will tell them exactly what they need to do to meet the standard. So it was a compromise that it would be a 2-part document. We'll get our guidance in as part 2, that's just informative, and the Europeans will have just the normative part of the section. So obviously they like to sell standards, so somebody came up with the bright idea like, hey, let's now merge it back to one like it was in the first place. So it was supposed to just be editorial changes, but like with any document that goes through review, everybody tries to get in their 2 cents worth, and a lot of technical comments and changes got put into it as well. Some of the key points is it does include healthcare now, so it actually does apply to hospital sterilization, although that's considered to be voluntary in the US, and because of all the requirements and the resources it would take to implement that, it's not likely that many hospitals will actually go through and validate for that, even though it's an option for that. It's considered more mandatory in Europe that healthcare facilities do have to go off of this standard. It also excludes prions or spongiform encephalopathies. That's like mad cow disease. It excludes that because there's really almost no type of sterilization that can get rid of those all the way. You can use some steam sterilization at very, very, very high temperatures, but ethylene oxide, radiation, some of those other ones will not destroy that. So that's one of the things that they excluded from the standard. and also gas in flexible chambers. Is anybody familiar with the Anderson process? It's basically a bag that you would seal your product in, and we have what they call, it almost looks like a grenade. It has a little glass canister of ethylene oxide inside this plastic housing. So you basically break that, through the packaging, you break that canister and it allows the ethylene oxide to release into a bag. That bag's then placed into an incubator, and then that's allowed to sit at an elevated, like at sterilization temperatures around 55 degrees for a period of like 16 hours. And the gas slowly, it's almost a semi-permeable bag, so it's almost non-permeable. So the gas slowly diffuses out of this bag over this 16 to 18 hour period. And so it excludes that. There's a separate AAMI document for this type of process. So this document does not apply to that type of ethylene oxide sterilization. And because of this standard change, the ISO 11135, a lot of the associated AAMI TIRs have also changed. TIR 14, which is contract sterilization. 15 is physical aspects of ethylene oxide sterilization. 16 is the microbiological aspects of ethylene oxide sterilization. And then TIR 28, which is, that is more like process equivalency and product adoption. So because ISO 11135 changed, all these will have to change as well. So the document's broken up into 12 different sections. They've organized it a lot better than it was in the previous version. So it goes through all these different ones, makes it a little easier to read. We'll go through some of the changes that happened to all of those. There's also a lot of different annexes. And these are some of them are guidance, some of them are informative where you don't have to meet. If you look at Annex C, the number of temperature sensors, BIs, things like that are required for your validation cycles, that's considered informative. It's not a requirement to meet those exactly, although most people will go off of that. There's a table in there that says if you have this much product volume that you're sterilizing, you have to use this many temperature probes, this many BIs. So most people still go off of that, even though it's informative. One of the biggest changes was Annex E, which is single lot release. This is for product that's made on an infrequent basis, or maybe it's a small lot to be used for clinical trials, that you can go through kind of a reduced validation so you can end up with sterile product without going through a full-blown validation. So it has an annex that covers that. Previously, that was covered in AAMI TIR 16, which is a US document. The Europeans were not big on batch release because there was not a European document that covered that. So this will help the batch release process move forward in Europe a little more. So again, some of the changes for it, the layout is improved. There's a lot of good, at the beginning of each section, there's kind of a definition of what this section's really talking about. So as a reader, of somebody that's not as familiar with ethylene oxide, it's going to help with that process as well. They changed some of the standards, they removed some of them. Not that they're not applicable, like ISO 10993-1, which is biocompatibility. It's not saying that you don't have to do biocompatibility. It's just they took it out of the scope of this document because there's other standards that you would clearly go off of. So they've updated some of the dates from some of the standards that's changed. Some of the definitions have changed. They've added some, they've removed some, and they changed some of the notes to it. So this is kind of a list of the ones. We're not going to go through, obviously, each one of these definitions, but they added quite a few. They also added, not only that we mentioned they added healthcare, they've also added reusable devices to this standard for devices that are going to be sterilized multiple times by maybe a healthcare facility. So a lot of these definitions, like reusable medical device, single-use medical device, some of those apply just to that to differentiate reusable versus single-use devices. So again, made some tweaks. Anytime they change standards, everybody's got to get their own definition in there. So they did change some of those. Again, healthcare facilities, that's what HCF, that's been put in there. So they put on the onus for the healthcare facilities' responsibility for validation and release of that. Even if they contract that out to like a third-party reprocessor, there's companies that some hospitals will send their devices to get reprocessed if the hospital doesn't do it. But this is basically saying it's still the hospital's responsibility for those devices. One of the key points for Section 5, which is the sterilizing agent characterization, this is looking at ethylene oxide itself. You don't have to really go through and do testing to characterize a sterilant as long as you're using a known blend or 100% ethylene oxide. If you were to use a different type of sterilant that hasn't been out there before, you would have to characterize it. But the biggest point to take away from this is that there's not a requirement to test raw materials, but you do have to test your finished device to see the effects of ethylene oxide. And so just on the finished device itself. A lot more information on health and safety. Occupational exposure to ethylene oxide is becoming a bigger deal. So there's a lot more safety put into this document that wasn't in there before. Section 6 talks about the process and the equipment characterization. This is where it describes the sterilizer. You're going to describe all the different steps of your cycle. It talks about transfer time. That's a new variable. That was not a variable in the old standard. Transfer time is the time it takes for moving the product out of the preconditioning room. And in a big contract sterilizer, they're going to do this with a forklift. They're going to take it out from preconditioning. It may take 20 to 30 minutes or more to load the new sterilizer, and that's considered transfer time, the time it takes to do that. And as you're doing that, that load loses temperature and loses humidity, which are critical components to EO sterilization. So that they added this as a variable that needs to be controlled. And so that wasn't in the old one as well. They also removed the steam quality and purity requirement. That applies more to steam sterilization. Steam's not really considered part of the sterilant for ethylene oxide. You do need the moisture, but they did remove the requirement that you don't have to meet pure steam like you would with a steam sterilization process. Then section 7 is the product definition. This is where you're looking at your product, you're describing it, you're looking at the microbiological quality, things like the bioburden on the product, the ability of whether it can be cleaned. These are all things that are in this section. They also added load configuration. Before it said the product and the packaging had to allow sterilant penetration, had to allow humidity into that. Now they're also saying that the load configuration itself, so the full load, whether it's pallets or boxes, you have to design that to help optimize the penetration into the load, because sometimes the load itself can make it very difficult to get that into. Transfer time, again, is another key factor in this section as well. And they also added a note about bacterial endotoxin. And that's more just a clarification. A lot of people that may not understand that for pyrogen testing or LAL testing, sterilization doesn't remove that pyrogenic material from the product. So even if it's sterile, that material is still present. And so it's just a note that it's not going to be destroyed by that. You still may need to go through and do pyrogen testing. Section 8's process definition. This is where you're really characterizing the cycle itself. You're looking at the different parameters that you're going to run and really specifying those. You have to do this on any new or modified product and document that. And what it says here is it says BIs must be placed in the appropriate PCD. It used to say the BIs had to be placed inside the product. Now it allows you to use process challenge devices. It clarifies that. You've always been able to use them, but it just more clarifies the fact that you don't always have to use your actual product. Section 9 then, that whole section deals with validation. This is kind of a no-brainer, but they put this in as a clarification that the protocol and procedures must be approved prior to starting your validation. I don't know that anyone that's ever done a validation that hasn't had their protocol approved ahead of time, but they felt the need to put that in there and highlight that. Again, more safety on that. It talks about the calibration confirmed as part of the IQ. And it doesn't allow any positive internal PCDs, although it does allow you to have some external BIs. If you've shown external BIs are basically placed on the outside of the load just to help the contract sterilizers with ease of removal. You don't have to tear your whole load apart to get in to remove the biological indicators. You can place them on the outside. But typically you have to have a biological indicator that's more resistant to put it on the outside, because it's going to be easier to sterilize just the outside surface of that load than it is down in the very center of it. So generally you have an even more overkill built in because you have an even harder one to put on the outside. So that's saying if that external one is just a worst case monitor, you are allowed to have some of those positive as long as the ones down inside the load are killed off. One of the things they changed to help incorporate sterilizers that just use conditioning only and not just preconditioning is that at the end of the conditioning time is when the sterilization load temperature and humidity have been established. So when you're doing ethylene oxide sterilization, normally you precondition it. A lot of times that's in an outside room where you heat up the product, you add moisture, which is going to help facilitate the sterilization. And then that's going to go into the chamber, and as you move it, we mentioned you lose some of that heat and humidity, and so you want to get that back in. So conditioning is where they add steam, which also brings back the heat and humidity into that load. That's allowed to sit for a little bit of time, usually like 60 minutes is pretty common, and then the gas is added there. But some cycles only use conditioning. They don't use outside preconditioning. So this change was done to allow that to go through for cycles that only use conditioning only. They've added requirements to document inert gas flushing, conditioning, and post-exposure flushing, requirements for that. That's the vacuums that you do at the end of the process to remove the gas. Sometimes it takes 40 minutes or more to pull the vacuums in some of these big chambers. And as you still have some gas inside the product, you're going to still continue to kill the product. So this is considered a variable on your process as well, and you want to characterize that and have that be as consistent as possible from cycle to cycle, because that can add to the amount of kill that you can get. Some key points: your dunnage load configuration should be representative of your actual product. This is an interesting one. I had one company that they didn't want to use their actual product, so they used simulated product, and it was probably the worst dunnage. Dunnage is basically just simulated product, something that you're going to use to simulate the density and the materials of your product. They used old clothes and wadded up coat hangers as it's probably the worst. I mean, there was actually pairs of pants and shirts that they were trying to simulate their product, so that wasn't very representative of their actual device, which was a plastic and metal device, and so they had to go back and come up with new dunnage that was more representative. So if you're going to reuse your loads, you have to aerate those between cycles. And so that's it for the validation part of it. Then you get into section 10, which is routine monitoring and control. This is where you're just going to do, once you've validated it, you're just going through cycles that are going to go through your routine production cycles from then forward. It talks about some of those new parameters that we talked about, the vacuum times, the time to evacuate. All these things need to be, you have to characterize that and record the data from it and retain that. Again, you want to make sure it's consistent, that the vacuum time is as close as possible from cycle to cycle. And then one big change, this affected a lot of people. A lot of times people would validate using one process challenge device that simulated their product, but then once they got into routine sterilization, they would just put a BI inside a pouch on the outside of the load and say that was just a catastrophic indicator, that they didn't need to use as hard a one as you would during validation, since the process was already validated. This is saying that if you're going to use a different BI than is used for your validation, it has to be at least as hard to kill as what you did for your validation. So you can't use those ones where you just put one on the outside of your pallet that's in a pouch. So product release, this is where these are the requirements to be able to release the product following sterilization. You can release the product, obviously, if none of the biological indicators grow, if you're not doing parametric release, or if all the data, the physical parameters have met the process specifications. And if you get a positive BI, you can't just release based on an acceptable product sterility test. Because when you say your product's sterile, it's got to be sterile to what they call a sterility assurance level of 10 to the minus 6. So that's basically saying 1 unit in a million can be non-sterile. So if you're doing a sterility test on product, you would have to test 1 million devices to be able to prove that. So you can't just say that I tested 10 samples and therefore my whole load is sterile. And so this is clarifying that aspect as well. And then maintaining process effectiveness, section 12, that's talking about revalidation or the extent that you do have to revalidate. You just have to review your validation data annually to see what extent you need to requalify. Generally with EO, you only do it every 2 years. And then in that off year, you can do what's called a paper validation. As long as nothing's changed, no adverse effects, then you don't have to do it every single year. So that's one of the big advantages. They do have this chart in there that goes through that process. If you made no changes at all, then and you've requalified within the last 2 years, then you go down to that bottom section where it just says document your rationale. If you've made some minor changes and you haven't documented or validated in the last 2 years, then you have to do a reduced qualification, which would consist of 1 half cycle instead of 3. Or if you made some significant changes, then you'd have to go through your entire validation all over again. So this decision tree kind of spells it out pretty clearly. It's a good change that they made to the standard. So that's it with the ISO 11135 document. That's the main document you use for ethylene oxide validations. The next thing we're going to talk about is an item that is causing issues in the industry right now, and that is gas mixtures. A lot of the gas mixtures, ethylene oxide, 100% ethylene oxide is flammable and explosive. So historically, a lot of hospitals and people that have small sterilizers in-house would use a gas blend, and this would be a mixture of ethylene oxide with something like Freon. Back in the day, they used Freon back in the 80s. And that was basically is just to make ethylene oxide non-flammable, so it reduced that explosion hazard. The problem with some of these, the CFCs and the HCFCs, is they were ozone depleting. So as part of the ban on a lot of these processes, because it can hurt the ozone layer, has nothing to do with ethylene oxide as a sterilant, but just these carrier gases that make it non-flammable also destroy the ozone layer, so they've been phased out. And as part of that phase out is part of the Clean Air Act. And so this caused an interesting issue with it. When they first changed, there was one called Oxyfume 2002, which was 10% EO. It was the first one to get phased out because it was more ozone depleting than some of these other mixtures. They did validations to show that when you switch from this version to another version of the mixture, that no change was seen. So you didn't see any issues at all. So the complete phase out of all the HCFC blends had to go in effect of January 1st of this year. So the problem with this is most of the BI manufacturers were using these gas blends just because they were non-flammable. But what they found is when they switched from this gas blend to 100% ethylene oxide, then you ended up with a lot of major differences between the kill rates. You got a lot more kill with 100% than you did with these gas blends. So it made the D values a lot lower, which sounds like a good thing, that you get more kill. But if you understand biological indicators, and these are what you monitor the process with, these are just where they put the spores onto either a paper carrier or some type of carrier material, and this is what you're gonna monitor each and every cycle with. When you look at how they're done, they have a defined resistance that they have to go through called D value. And what D value is, it's basically the time it takes to reduce a population of the spores by 1 log. So you go from 1 million to 100,000. It's how long did it take to do that? And from 100,000 down to 10,000, how long did it take that? That D value is just a judge of the resistance of that. It's just the time it takes to reduce the population by 90% or 1 log, which mathematically those are the same. And so the higher the D value, the more resistant it is. So every time you buy biological indicators, they have a cert that comes with those. And so when they tested these, they found that the D value on these, they did a round robin study with multiple BI manufacturers, and they found that there was, in some cases, up to a 40% decrease in the D value. And so again, that sounds like that might not be a bad thing, that you're getting more kill than you did before. But the problem is a lot of the standards have requirements for what that D value has to be. So all the D values that you get come with a range, like on this cert here. In that particular example, that D value is 3.2 minutes. That's saying it takes 3.2 minutes to reduce the population from 10 to the 6th down to 10 to the 5th. Just kind of a judge of that. And what the standard says, if you look at USP, which is one of the biological indicator standards, there's several out there, but it had a range that it had to be at least 2.5 to 5.8 minutes. And then ISO 11138 said it couldn't be less than 2.5 minutes. Same thing with the European Pharmacopoeia. Couldn't be less than 2.5 minutes. So now if you're getting 40% more kill, these D values are going to look like they were lower than before. So they may not meet the standard. So it's more of a regulatory issue. The problem is the BIs themselves have not changed. You're using the exact same BI you used 5 years ago, 1 year ago. It's just the measuring stick that you're using to monitor those has changed, so it looks like they're different. So that created the industry choice. Yes, go ahead, question? Yeah, generally if you validate a BI, you're sort of stuck with that one. There's no requirement that if you do change BIs that you have to go through a whole new validation again. A lot of people will, but because these standards have requirements of what they have to meet, there's generally more difference from the same BI manufacturer, say Steris, from one lot of their BIs to the next lot of their BIs. There may be more difference there than maybe from going from like a Steris BI to a 3M BI. And so the organism is the same, and the resistance requirements, population requirements are generally the same window they have to meet. So technically, scientifically, you probably wouldn't need to validate that again, but regulatory-wise, a lot of people do if they do switch BIs. So again, the BI itself did not change. It's the same organism, same thing it always has been, but now it suddenly looks like it's a lot lower, a lot less resistant. And so, yes. The question is, why did the industry create a minimum requirement for the D value? And the real reason for that is they wanted to try to make these as consistent, because you have so many different manufacturers of BIs that they wanted them all to be as consistent as possible. So they set both population standards as well as resistance standards so that everybody kind of is giving the same product. So if you used one BI, it wouldn't be radically different from another one. So good question. So again, the BI didn't change here, just the carrier gas, the 100% gas gave more kill. So the choice in the industry was they either have to make the BIs more resistant, just so that when it does reduce it by 40% in some cases, that it's still going to meet the standard. But can you see a problem with that? If you now suddenly make your BI way more resistant than it used to be, just so it meets this 2.5, you're using a BI that's harder than it used to be. And there's a lot of people that their cycle might have been just on the edge, that it was just giving you the amount of kill to get there. So suddenly if your BI is 40%, or your cycle, BI is 40% more resistant than what you used before, you could end up with sterilization failures when there's really no need for that. And so then the more smart choice was change the standards to justify that this is due to this change in the different types of gas. And so the AAMI committee just voted on this, and they voted 14-4 to end up what their recommendation is going to be to change the standard to add a statement that says if you're using 100% EO, the value would drop to 2.0 minutes, but if you're using any kind of other type of mixture, then it would be stay at 2.5. Which makes sense. A lot of people are kind of pushing back on that a little bit because they've put it in their 510K filings or their PMA. They actually specify to that detail that they use a certain BI that has a certain D value, and if they change that, then they're going to be out of compliance with that. But to me, that's a lot better choice than making the BI artificially harder than it was before just to meet this specification when there's a clear reason why that happened. So that's the dilemma that the industry is faced with, and so still in process. That needs to be presented to the ISO committee. So we'll see how that plays out. So a lot of you have probably heard about the flexible endoscope issue that's going on. This is just slightly related to ethylene oxide. Basically, these duodenoscopes, they're flexible lighted tubes. They're threaded through the mouth. They're really used to drain fluid from the pancreatic or bile duct. So this issue has not really happened with all endoscopes. It's just usually these ones for duodenoscopes has been the problem. So every once in a while, there's hospital-acquired infections that pop up. They have sometimes they'll have these little clusters of outbreaks. This one was a common one that happened. But the problem was it's a very resistant organism that's resistant to the last line of defense. It's an antibiotic that is carbapenem that is very, very resistant. And this antibiotic basically is the one they use when nothing else works. Unfortunately, some of these organisms have developed resistance to that, and that's a big issue because now you have an organism that almost no antibiotic will work against. So this got a lot of media attention because a few patients died. I think UCLA, there was 2 patients that died because of this, and so it's been in the media a lot. It's been a big issue for that. So the big deal with these, like I said, no antibiotics can help for this. So some of these infections can have a mortality rate up to 50%. So it's definitely something you don't want to get if you go to the hospital. But it's important to know that these infections are not just caused by this type of device. This is just the most recent one that came out. These are devices that are cleaned and then re-sterilized and used on the next patient. So they use them on one patient, they reprocess them, use the same device on the next patient. They're very expensive. Some cases they're $40,000, $50,000 a unit. So obviously they can't make them single-use devices. So they have to be cleaned and re-sterilized. And so what they found is some of these are very difficult to clean, and that's where they found out. This is a picture of these Petri dishes here. This is how they do antibiotic potency. So you can see on the one on the right, some of these antibiotics are having zero effect against that microorganism. So where it has an effect, we see that area where it's clear around that, that's showing that organism's susceptible to that type of antibiotic. So the problem with these organisms is they're very much not affected by antibiotics. But the incidence is quite low. There's about 500,000 of these procedures done every year, and there's only been about 100 cases of this CRE, or these very resistant Enterobacteriaceae. That's the organism that causes that. There's only been about 100 cases over the past few years, so it's not a huge volume-wise. It's not that the cases are going up, it's more that the severity of them, because they are antibiotic resistant, causes such a big issue. And so they're looking at trying to clean and find better ways to do this, either redesign the devices. So some hospitals have gone out on their own to sterilize these using ethylene oxide. It's something that originally they're just designed to be disinfected in the hospital, not necessarily sterilized. But some hospitals, because of this, have gone to using ethylene oxide to sterilize it. So it'll be interesting to see if the industry follows suit with that and continues on with that. But since these devices, they're not really made to be, they weren't designed with ethylene oxide sterilization in mind. So these manufacturers that have to go back and do testing to see, can they be redone X number of times? Because these are used over and over and over again. And so the manufacturers are working with the FDA, the CDC, and industry trying to figure out solutions for this issue. Some of them is a surveillance program where they would use the device, clean it, reprocess it, and then they would do a screening test where they would look for this organism. And in some cases, the device is held in quarantine until that surveillance comes back and they say, okay, there's none of these organisms on there. So that's one option. Some of them do it between every single use. Other ones may only do it weekly, maybe on Monday mornings after the device has been used for a full week. So some hospitals are doing it differently. Some of these scope manufacturers are redesigning the device, changing their cleaning and disinfection procedures. Sterilization will be interesting to see, because sterilization would be, in my mind, a great option, because that way you don't have to worry about that. It's going to give you that added safety factor to that. And in some cases, they're even looking at making maybe single-use ones. So that's kind of a big industry. A lot of you have probably heard in the news, a big issue that's going on right now. The last one is a list of all these EO standards that are changing. We mentioned ISO 11135 changed. So all these related AAMI TIRs have also being revised as we speak. The revision process can take 1 to 2 years. It's a long, complicated process to get these things done. So a lot of these, you'll see changes to these AAMI TIRs. TIRs are technical information reports. These are basically guidance documents to show how you need to meet ISO 11135, which is kind of the parent document. The BI standard, ISO 11138, these are the BI manufacturer standards. These are changing as well. We talked about that issue with the 100% ethylene oxide versus the blend, about the difference in the D values. There's a proposal by the US to put this in that standard to explain that difference. And then they removed the requirement for D value testing at 30 degrees. It used to be you tested it at 54 degrees and then also at 30 degrees. So they're removing that 30 degree requirement. USP has the revision, the biological indicator section on that is being revised as well. It used to have kind of a complicated scheme that said it couldn't be more than 0.48 logs or fall under 0.3 logs of what the labeled population is, which that sounds very confusing. They've changed that now to say the population can't fall under 50% or be more than 300% of what the labeled population is. And so that matches what ISO 11138 states. So USP is kind of in revision as well. There's a growing flammability issue with ethylene oxide sterilization. As devices become more and more complex, more and more automated, there's a lot of battery-powered devices. Again, since 100%, and 100% ethylene oxide we mentioned is explosive and flammable, it's used, there's only one contract sterilizer I know of that uses a gas blend. Do you guys use a mixture, use 100%? 100%, so almost every contract sterilizer, really from a cost basis, 100% is cheaper than those gas blends. There's a company in California called Parter Medical that uses a gas blend, but they're basically the only one. So because 100% is flammable and explosive, obviously anything that's an ignition source, there has been a series of, not a series, but there's been throughout the decades that ethylene oxide has been used, there's been explosions in the industry in different sterilization chambers or oxidizers, the systems that use to get rid of the ethylene oxide. And so flammability or any kind of ignition source in an explosive environment, obviously not a good thing. So the contract sterilizers are definitely wanting to know if you have anything that's got stored energy or batteries, that's anything like that. EOSA, which is the Ethylene Oxide Sterilization Association, has come out with some guidance. Some contract sterilizers will actually refuse to use any devices with batteries. Some of them will help doing a safety assessment and see if you need to do that. Here's some examples of that. Pen lights, irrigation pumps, powered staplers, anything that's powered with batteries, cochlear implants, devices with motors where some kind of charge can be stored up, powered screwdrivers, things like that. So these are all things that potentially can be an ignition source in this flammable environment. So a lot of times they look at the voltage and the amperage to see if that combination can create it to make it over the flammability area. Is there multiple safeguards? One safeguard might be that you have an on and off switch. Problem is that switch maybe during shipping gets bumped and it can get turned on. So that can be an issue. So they want to have multiple safeguards. Like maybe in addition to the switch, you also have a tab on the battery that even if that switch gets turned, then that battery's not connected and can't complete the circuit. And so they want to know if the device somehow inadvertently got damaged during shipping. If the device is damaged, is there an issue that it could malfunction and cause any kind of spark? So this is an example of a checklist that's in that EOSA document. Ethylene Oxide Sterilization Association, again, it kind of walks you through that process. It has certain things like if it's more than 5 amps at 12 volts, that's basically, it says you cannot sterilize with ethylene oxide because of the flammability potential. If it can activate and come on during sterilization, obviously you don't want to do that. So there's certain things that are kind of, there's no way you're going to be able to process it if you check yes to certain items on that. But it does have a good way to walk you through that. So that's another industry that we see, this flammability issue that continues to grow as devices become more complex. So that's just kind of in a nutshell some of the changes to the standards and some of the issues that the industry is facing in regards to ethylene oxide sterilization. So thank you guys for coming, and I hope you enjoy the show. And I hope you wore comfortable shoes for all the walking that you're doing. So thank you.